Hi everyone. This video will describe the activation of the humoral immune response in detail. The activation of the humoral immune response always starts with the B cell and also the antigen presenting cell. Step number one consists of two parts. The first part that involves the phagocytosis of the pathogen and also the activation of the B cell. Both of these parts will occur at the same time. So we will look at the first part. When there's a pathogen that carrying the antigen, the phagocyte, for example, like the dendritic cell and also the macrophage, will go and engulf these pathogen. So after they engulf the pathogen, they will digest the pathogen and extract the antigen. So later on, these phagocytes will present the antigen on the surface through the class 2 MHC molecule. So that means that these phagocytes will later on act as the antigen presenting cell to present these antigen of the pathogen on the class 2 MHC to the helper T cell. So the helper T cell with the specific receptor will recognize this antigen that is being presented on the class 2 MHC. And once the helper T cell recognize the antigen, they will be activated and undergo the coronal selection. So they will do the mitosis to produce two types of the helper T cell. The first type is the memory helper T cell, which will play a role in the immunological memory in future. While another type would be the effector cell, which is the activated helper T cell. So at the same time, the same pathogen with the same antigen will be binding to the antibody on the B cell. And this antibody is normally the IgD or the B cell receptor. So the antigen will bind to the antibody on the B cell and thus activate the B cell. Then after this, when we have the activated B cell and also the activated helper T cell, then next we will move on to the step number 2. So in step number 2, the activated B cell and also the activated helper T cell will go to find each other. They will come close to each other, and these activated B cell, they will start to act as the antigen presenting cell to present the antigen to the activated helper T cell. These B cell, after they bind with the antigen, they will go through a process called internalization in order to present the antigen on the surface of their cell. And since this B cell can act as the antigen presenting cell, so that means that this B cell will also have the class 2 MHC molecule on their surface. So this antigen will be presented on the class 2 MHC of the B cell. So the activated helper T cell will recognize the antigen that is being presented by this activated B cell, and after that, it is going to release the cytokine. So this cytokine is going to fully activate the B cell, and this B cell will start to undergo coronal selection. So same thing again, the B cell will undergo mitosis to produce two types of the cell. So one type would be the memory B cell, which is important for the immunological memory, while another type would be the effector cell, which is the plasma cell. Then next, we will move into the step number 3, where the plasma cell will start to secrete the antibody. Then after the antibody is being secreted, then we will move on to step number 4. So this antibody will go and bind to the antigen that occurs on this pathogen. So with that, the antibody will either neutralize it or cause the agglutination of this pathogen, and thus leading to the elimination of this pathogen. So with that, that is basically how the humoral immune response being activated and act on the pathogen.